Alright guys, welcome back. So we're going to continue discussing our progression of how we believe life on Earth got started. And so last time we were talking about just the conditions of early Earth, it being so hot, not a lot of water on the surface until after about 3.9 billion years. We talked a little bit about protocells, those really early beginnings to cells with just sort of this lipid membrane and some simple metabolism, RNA being the first genetic material. And so today we're going to pick up talking about how do we get those early protocells to finally start looking like something living. So we know based on our geologic fossil record that prokaryotes come first. So if we remember your prokaryotes, these guys are your simple cells. They don't contain any nuclei, there's no mitochondria, there's no real internal structure to them. But they are now going to meet all those characteristics of life. So metabolism, replication, similar molecules, so on and so forth. And so those early protocells eventually get replaced by these first prokaryotes. And prokaryotes are pretty much the only living organisms, simple-celled organisms, that are going to inhabit Earth from at least 3.5 to 2.1 billion years ago. And we can see this in the fossil record. So something to keep in mind in terms of classification, we've got two major domains out of the three total we have. They're all prokaryotes. So we talk about your eubacteria being one domain, archaeobacteria being a second domain, and then that third one eventually will be made up of your eukaryotic cells that we'll talk about how they get, how they end up evolving in just a little while. So big difference, don't get stuck in this, but just something to be aware of. What's the difference between eubacteria and archaeobacteria? It really comes down to their cell wall structure. Eubacteria contain this sort of sugary polysaccharide compound called peptidoglycan. Archaeobacteria do not have them, and instead their cell membrane is a little bit more lipid-based. In terms of where these guys live, even currently, archaeobacteria are also sort of thought of as extremophiles, so these are ones we tend to find in really hot places or very salty places on Earth, where your eubacteria are sort of your more quintessential, traditional, what you'd think of as bacterial cells or prokaryotic cells. That being said, evolutionarily, actually, the archaea we historically thought were much older, hence archaea, like archaic, ancient. That's where the name came from, and yet we actually found out as we started to do some more DNA testing and RNA testing that actually the archaea are a little more like our current eukaryotic cells than the eubacteria are, which is sort of an interesting piece of information that'll actually come back up when we get to the last part of this unit. But what do we know about the evolution of prokaryotes? Well, besides the fact that they are the sole inhabitants initially in terms of living things on Earth, they're ultimately also going to be responsible for getting us enough oxygen in the atmosphere to really start helping life take off. So we know that looking at early Earth atmosphere, there is next to no oxygen present at all. And yet, what's one of the first things we think about as a necessity to most living things is the presence of oxygen. So where is it ultimately going to come from? Well, we're going to have to evolve that process of photosynthesis. Most of those early prokaryotes that are hanging around back here in this 3.5 to 2.1 billion years, most of them are heterotrophs. So they're out just sort of consuming other organisms and doing some really simple metabolism that way. They're maybe chemosynthetic, right? So using chemicals to make their own food. Photosynthesis doesn't seem to show up until somewhere on the order of about 2.7 to 2.3 billion years ago. And how we know that that photosynthetic process finally evolves is because we get this whole part in our geologic record that we talk about as the oxygen revolution. So when we look at the layers of rock in our fossil record, we can see a distinct line of rust. And rust is iron oxide, so it's iron plus oxygen. And there's a very distinct layer where it shows up, and it then exists above that, but below that doesn't exist at all. So we know somewhere in this band that we're getting that early photosynthetic process to happen. What's one of its major byproducts? Oxygen. And so as we continue to see these bacteria go through and start using photosynthesis, producing oxygen as a byproduct, it starts building up in the oceans, all right, which is then going to combine with all that iron, which settles out as that rust at the bottom of the ocean. That's where that initial layer starts coming from. And eventually, so much oxygen is in the oceans that it's combined with so much of the iron that there's nothing left for it to combine with, and so instead, it starts gassing out into the atmosphere. Ultimately, that's going to allow our ozone layer to form, which is really important, because that ozone layer is O3, so oxygen, 3 atoms of it, right? And it's now going to provide that protection for any life that could potentially get onto land. Before the oxygen revolution, life couldn't exist above the oceans. The only thing that was keeping them protected was the water itself. Well, now that the oxygen revolution comes around, that ozone layer forms. Well, now we have this extra external atmospheric layer of protection, so eventually life is going to be able to get out of the oceans because of this. It's also going to allow for the evolution of cellular respiration, and something we'll talk way more in depth about later this year, is 2 major processes that cells could utilize in order to make their own ATP, or energy molecules, right? So those really early cells, those really early prokaryotes, before this oxygen revolution, pretty much had one option, and that's fermentation. Problem is that the fermentation process, as we'll see later this year, can only get you 2 ATP molecules for every sugar you put in. Not particularly efficient, but I can do fermentation without oxygen. Now that the oxygen revolution has occurred, now I can get the evolution of cellular respiration to happen, and that becomes a big deal because now I can take that same sugar molecule in that cell and now make somewhere closer to 36 ATPs for every sugar. Way more efficient. I'm getting way more energy out of the same amount of food that I have to take in. And so all of this is going to set up a really interesting dynamic to getting more complex cells, bigger organisms, multicellular organisms, and eventually getting a cell to land. So this oxygen revolution really sets up so many parameters that is going to allow life to start evolving beyond just these single-celled, simple organisms. So here's where I mentioned the evidence, right? How do we know that that oxygen revolution happened? We can see that rust accumulate in our sediments in about 2.7 billion years ago is sort of where that first layer really shows up. And once all of that iron had kind of fallen out of solution in the oceans because it was now connected to oxygen, it eventually started gassing out of the water. All right, we get these oxygen bubbles that burst, and then that oxygen ends up in the atmosphere. And so that tells us that the cyanobacteria, which are sort of our biggest class of photosynthetic bacteria still existing, they must have originated, at least their ancestors must have originated as early as 3.5 billion years in order to have enough time for that iron to sort of settle out. So now I've got photosynthesis in place, I've got an ozone layer in place, I have the potential for cellular respiration to be in place, And now we can start talking about, well, how do I get from these really simplistic prokaryotes to the more complex eukaryotic cells that we know exist today? So the evolution of those eukaryotes we can see based on fossil data go back to about 2.1 billion years. Okay. So how do I go from all of this lack of internal structure to all of this? Well, there's a theory out there called the endosymbiont theory. Okay. And we're going to focus on it in terms of just the mitochondria and chloroplast, although the theory itself encompasses a lot of that other internal structure, the nucleus, as well as other organelles. But for our purposes, we'll simplify it a little and just stick to the evidence for the chloroplast and mitochondria. And so if I break this whole idea apart, right, it's an endosymbiont. So endo, if we know our prefixes, or starting to learn them as we should at this point, that endo means within. And symbiont, we're talking symbiotic relationships, which is something you should have come across in the past as well, where I've got some relationship with one organism depending on another and vice versa for something. In this case, we're really looking at what is probably more appropriately connected as a mutualistic relationship, where we're going to get one cell that ends up living within another host cell, and eventually that connection becomes so tight between the two of them in terms of their interdependence on each other to survive that we can't separate them anymore. So we believe that ancestors of those mitochondria were once free-living prokaryotes, specifically aerobic, so they required oxygen, and they were heterotrophic, so they needed to consume something in order to make their energy, and they were once free-living prokaryotes, chloroplasts, similar idea, but they would have been photosynthetic prokaryotes, and they ended up getting eaten by these bigger cells. Alright. So they end up gaining entry into a bigger host cell by being eaten via a process called endocytosis, which really is just one cell taking its cell membrane, surrounding some sort of food particle, and bringing it into its internals. And for most cells, they're going to then break down that prey molecule, that prey cell, use the molecules from that prey, just like we would do if we ate any sort of food. But for whatever reason, in this particular situation, those particular prey, those current mitochondria, current chloroplasts, never got digested. And so instead, we ended up setting up this symbiotic relationship where both the big host cell that had eaten the little one now gets this extra metabolic process it didn't have before, whether it's the cellular respiration we're going to ultimately talk about with mitochondria, or the photosynthesis with the chloroplasts, or potentially both. And that big host cell now gets more efficient metabolism, and that little cell is now protected, alright, and gets everything that it needs from the bigger host cell. So over time, and we're talking long periods of time here, that process continued to sort of happen sporadically. Those cells ended up being interdependent, and now they have become a single organism to the point where I can't remove a mitochondria or a chloroplast from a cell and have it function on its own. It just no longer is capable of doing that. So this sounds all cool and weird and maybe sci-fi-ish, so where's the evidence for it? And you definitely want to make sure that you can cite some of this evidence for this endosymbiotic theory. One of the first ones is that mitochondria and chloroplasts both have a double membrane system. Well, where did the double membrane come from? Well, if I take a look at these guys, that inner membrane would have been its original cell membrane as a free-living prokaryote. That outer membrane came when the host enveloped it. Alright, something we'll talk about as a vesicle membrane, or a food vacuole, when we get to cell structure, probably about December or so. So those guys get taken in, and they now have an extra membrane on the outside because it's what the bigger host cell used to take it in the first place. Speaking of those membranes, well, if we go and dig into the inner membrane itself, which would have been the prokaryote's original cell as a free-living prokaryote, enzymes, transport systems, they're all homologous or very similar in terms of the way that they work and their structure to those kinds of systems that we currently see even in cell membranes, plasma membranes of current living prokaryotes, which is pretty cool. So there's a definite connection going on between those. Other interesting thing, bacteria split or reproduce via a process called binary fission. It's very simple cell division. And we can actually witness mitochondria and chloroplasts doing that replication process, that similar binary fission process within a functional cell, even when that bigger cell isn't dividing itself. So if I'm a muscle cell and I am in a human who is now training harder for, say, some big marathon, alright, my job is to make that human muscle cell energy. Well, the more and more that human cell demands that energy, I may need to make more mitochondria in order to meet that energy demand. Well, I can do that. I can replicate myself as a mitochondria and make more of myself within that cell and not need the actual muscle cell itself to reproduce, which is kind of cool. These guys also have their own singular circular DNA molecule that looks a lot like genetic material we see in prokaryotes. Okay? So actually, all of your mitochondrial DNA actually comes from your mom. Alright, she's the one who gives you your first cell. Not only does she give you half your actual nuclear DNA, but she gives you your first mitochondria. She gives you your first ribosomes. And so we can actually use mitochondrial DNA to trace maternal lines, which, especially from evolutionary standpoints, actually makes it a little cleaner. Because we're not having to sort out, well, what was mom's DNA versus dad's DNA. We know this was all maternal, which is kind of cool. They also have ribosomes. Similar to the ribosome structure of a prokaryote, every organism, whether it's prokaryote or eukaryote, has to be able to make DNA in proteins, use that DNA code to ultimately create proteins. And so having that ribosome structure within these mitochondria and chloroplasts once again gives us just a little bit more support to suggest that these guys used to be free-living cells. Okay, so I now have prokaryotes, eukaryotes have evolved, I've got photosynthesis, I've got cellular respiration in place, the ozone layer is now in place to protect us, and now we can finally start talking about getting out of the oceans and onto land. So we know that things like simple cyanobacteria, other photosynthetic prokaryotes, are going to be the first organisms that are going to start colonizing the land right at the coast. They're going to start coating those terrestrial or land surfaces way over 1 billion years ago. Eventually, multicellularity has to come about, so organisms with multi-cells, and true, what we think about as living things, fungus, plants, animals, they're not going to get out of the oceans until about 500 million years. But none of this would be possible without that initial photosynthesis evolving, getting us that ozone layer, getting us that protection, because without it, we'd get on land, we'd basically get irradiated. Our DNA would get blown to bits. Alright? Which is why everything stayed in the ocean before that, because the water was able to provide that level of protection against that radiation. Now that the ozone layer is in place, not as much of a concern, so we can get up on land without having to worry about that process. So, that being said, we're talking a difference between 1 billion years with prokaryotes on land to 500 million years before we really start getting to see a lot of those other, what we think about as life, plants, bigger animals on land. And there's really a big transitional period beyond just having that ozone layer and protection in place that's going to have to happen in order to get organisms out of the water and really get far up onto land outside of just being on the coasts. So those transitional adaptations have to come about in a couple of different ways. Well, one thing, if I'm constantly an organism surrounded in water, I don't have to worry about dehydration. So waxy coatings and oils and skin in that sense has to have evolved to prevent that dehydration. I also have to have a way to reproduce on land, which means internal fertilization has to evolve. If we're talking living in oceans and constantly being surrounded by water, think of fish. Most of them, they're not going and having internal fertilization. There is no sort of sex for them. They just spew out eggs and sperm because it's in a liquid. Those eggs and sperm will eventually find each other and fertilize, and off go new offspring. Well, I'm not going to be able to do that on land. I need that liquid material to still exist in order for sperm to swim, so we've got to have the evolution of internal fertilization come about. In terms of plants, they need some sort of vascular tissue, veins that can carry water from their roots up to their leaves and other tissues. That's not going to show up until about 420 million years. We're going to have a lot of associations or symbiotic relationships that are going to have to evolve to ultimately support life on land. One being these mycorrhizae, these plants and fungi that have these associations in the roots, and that's going to help those plants get nutrients and water out of the soil more quickly, more readily, more in a form that they can use. It's not going to happen overnight, so that evolution of that connection has to happen. And for the most part, arthropods, so things like insects, tetrapods are going to be the most widely spread and most diverse land animals that we see. And interestingly enough, tetrapods, so four-limbed organisms, we can actually see transitional fossils of them evolving from these sort of lobe-finned fish about 365 million years ago, which is kind of cool. And there will be an article you'll take a look at that'll also, we'll talk about them a little later as well.